Good afternoon. Thank you so much for joining today's GFRA webinar. Today, it is my pleasure to introduce Dr. Sandra Blaise-Boisseau. She's a distinguished virologist with over a decade of experience specializing in foot-and-mouth disease. Since 2009, she has been a key member of the Virology Joint Research Unit at the ANSES Laboratory for Animal Health in Maisons-Alfort, France. As the deputy head of the BioPIC team, Dr. Blaise-Boisseau plays an important role in managing the National Reference Laboratory for FMD, as well as serving as a WOA Reference Laboratory, FAO Collaborating Center, and EU Reference Laboratory for FMD. Her research primarily focuses on the interactions between host and FMDV, with the goal of elucidating the mechanisms behind FMDV persistence in ruminants. Through these innovative studies, Dr. Blaise-Boisseau aims to enhance our understanding of FMD and contribute to the development of effective control strategies. Today, the title of her talk is Molecular Interplay of FMDV with Type 1 Interferon Response and the Importance of the Model and Potential Links with Viral Persistence. Without further ado, Dr. Blaise-Boisseau, the floor is yours.
Thank you so much.
Do we have the presentation, Kayla?
I think Dr. Blaise-Boisseau, are you able to share your screen with the presentation?
Okay, sorry. Okay.
Perfect, thank you.
Okay, so good morning, good afternoon, or good evening, everyone. And first of all, I would like to thank the GFRA and organizer for inviting me to give this webinar for December 2024, namely Melanie, Giselle, Kaya, and Tessa for their support. And today it's my pleasure to present the work carried out in our team at ANSES, so in Maisons-Alfort, close to Paris, but a work done with their partners, collaborators in the world. And so it will be about this molecular interplace of FMDV with the type 1 interferon response and the potential links with viral persistence. Okay, so just to start with some words to remind that to retain fitness and host adaptation, viruses have evolved multiple strategies in order to manipulate, hijack host machine to their benefits and to circumvent the host antiviral responses. And so, why studying host FMDV interactions? So, to better understand the FMDV pathogenesis, which includes to better understand FMDV persistent infection and underlying mechanisms, with the aim to design effective vaccines or drugs to prevent, control, and eradicate FMD. So definitely FMDV persistence does matter. This subject is controversy since its first description in the late 50s. It was by Van Beckom and collaborators, and this team showed that this prolonged subclinical phase of FMDV infections concerns over 50% of ruminants that can become healthy carriers, independently of the vaccinal status, of the vaccine status, and with a duration variable according to the species. And originally, the definition of the carrier status was set arbitrarily at 28 days post inoculation, post-infection. But in 2016, our colleagues from the Jonathan Hart's team show that they demonstrated that carrier status could be defined as early as 10 days post-infection in vaccinated cattle or 21 days in non-vaccinated cattle. And they defined a sort of transitional phase of the infection. And in this phase represents the period during which the clearance of infection can occur. Other results reported that definitely the viral persistence is not described in swine. And why FMDV persistence does matter also related to the potential emerging source of new recombinants that carrier animals can represent, as exemplified in these two papers. And definitely, the risk of transmission from carrier animals cannot be neglected, even if it can be considered as low, and it remains an obstacle to control and trade. And even after almost 50 years after the first description, this persistence remains an enigma. And the underlying mechanisms remains to be clearly identified, elucidated. So during all these years, some clues have been accumulated thanks to different works in order to elucidate such mechanisms. So first work were done using some cell lines or primary cells to show that there is, during this persistence, a co-evolution, co-adaptation between FMDV and host. A modulation of the immunity was also described in vivo in cattle. We also show and characterize transcriptional responses during FMDV infection in the in vitro, in the primary bovine cells. And some works have also shown and inferred some mechanisms for persistence according to some differentially expressed gene in nasopharyngeal mucosa, and with a potential explanation of persistence that occurs in ruminants but not in pig. And we also give our contribution to this building, and in the framework of the Transcriptovac project, it started in 2015 in collaboration with Swedish colleagues and colleagues at FLI. We were able to establish a model of persistent infection using primary bovine nasopharyngeal epithelial cells cultivated and grown under air liquid interface as multi-layer. And we also performed the transcriptional analysis of the host response in this model using O-type virus. And we were able to establish a new in vitro model from a pertinent species. We also compute two distinct transcriptomic signatures during acute versus persistent infection. And we also show that an innate antiviral response remains during the persistent phase, but diminished and ineffective at eliminating the virus. We also pointed out some interesting deregulated genes during these persistent infections as potential candidate host marker for persistent diagnosis. So just a small cartoon to recap on these signatures, transcriptomic signatures after analysis of these primary epithelial cells derived from the nasopharynx of bovine. And so the question, the hypothesis was that a sustained but diminished innate antiviral response, ineffective in eliminating the virus, would contribute to the balance maintained between viral presence and host response, and thus to FMDV persistence. And the question was, what are the FMDV host interactions that modulate the innate response to allow FMDV to establish and or maintain persistence? Just before to go on with this work, just a small a small slide about the type 1 interferon signaling network. It is an essential component of the innate antiviral response. It is the first feature of infectious agents. Schematically, it can be cut in two phases. The first phase of induction that starts with the sensing of viral of pathogen motif, and that lead to the activation of transcription factors, so IF3 and 7, leading to the production of interferon. And this interferon will link to the receptor, and by paracrine and autocrine signaling, they will, through the JAK-STAT pathway activation, it will lead to the production of many antiviral proteins encoded by the so-called RHG for interferon-stimulated genes. So this response is a sentinel response. It is the early detection, important for disease control. It prepares the adaptive response, so through type 1 interferons and pro-inflammatory cytokines. And if we look at FMDV, so our favorite virus, what was in the literature so far, you can see on this graph that many viral proteins were reported to be able to hijack this response, and through various antagonism strategies like interference with viral RNA detection, targeting signal transduction proteins, through degradation of transcription factors, so IL-3 and 7, counteraction of the JAK-STAT pathway, inhibition of host translation factors, negative regulation of IgG transcription, dealing with the Golgi molecular transport, and the degradation of antiviral molecules like PKR. So if we recap on that, what are the outlines? So we observe that FMDV dedicates almost its entire small genome to counteract the type 1 interferon response. Some host proteins are targeted by a single viral one. Others are targeted by several FMDV proteins, namely viral RNA sensors. FMDV viral proteins involve various mechanisms like proteolytic cleavage, inhibitory binding, inhibition of mRNA transcription levels, debiquitination, as well as unknown mechanisms. Host proteins are the upstream phase of the induction of the interferon pathway are the main targets, and also main targets for other piconaviruses like EMC or enteroviruses, and proving their importance in setting up the innate response. But more than 50% of these interactions were described so far and demonstrated by overexpression of proteins from species not susceptible to FMDV, so mainly human proteins. So the question are, do these interactions exist in the natural host? Is there a conservation of this PPI, so protein-protein interaction between species? Which interactions during acute versus persistent FMDV infections? And we can also add this paper, it was published a couple of weeks, about another antagonism, and it was not expected to have this kind of antagonism because it targets the C-GAT-STING DNA sensing pathway. So at the beginning, a pathway dedicated to the sensing of DNA motif pattern, and but this pathway, it was shown that multiple families of single-stranded positive RNA viruses like FMDV are affected by, and in turn have mechanisms to inhibit this pathway. And it was reported for the 2B, and in this case, it is not at the protein level, but at the transcription level. So many mechanisms against, so viruses are very inventive to meet the goal to hijack the cell. And so it's now after this introduction and this recap on the interferon response and the relationship between FMDV and the type 1 interferon response, and I go back to this history after the transcriptovac project, and as I showed in a couple of slides earlier, about this transcriptomic response we observe with this diminished and ineffective interferon response during the persistent phase. And these results lead us to two projects. So a first project that I wrote, it was a PhD project called PERSIFA, and the aim was to study FMDV persistence and innate antiviral response, and particularly to study various host molecular interaction using interferotomic approach in 4 target species, and was done by my former student Morgan Sari. And in 2020, I also applied for an ICRAC project called FMDV Persistomics, still in collaboration with Michael Eschbaumer at FLI, Jean-François Valarcher from the SLU in Sweden, Sion Sano in Belgium, and colleagues from the Shab Institute in Ankara. And in this project, we had 3 main work packages to serve the goal. The goal was to go from the proteogenomic host response signatures until diagnostic of markers and therapeutic control of persistence. So 3 work package. So the first one dedicated to the host response in FMDV persistent infection, the second one dedicated to markers of FMDV persistence, and the third one about the modulation of FMDV persistence. And I will continue only with the work package 1 and what we show, what we study and found for this concerning the host response during FMDV persistent infection ex vivo or in vitro. So to be more precise, our aims for this study was to uncover alterations of host response during persistent FMDV infection of cattle. And we had in mind that one of the major viral proteins known to be involved in host innate response evasion during acute infection is the leader protease L-Pro. And this leader protease L-Pro is known to inhibit host cell mRNA translation and to block the type 1 interferon response. And leaderless FMDV variants of serotype A have been shown earlier to be strongly attenuated in vivo, with the potential, but the potential for persistent infection was not studied and remained unknown. So our question was, could leaderless FMDV establish a persistent infection? So the first work was to produce by reverse genetic the required viruses, and it was done by our colleague at FLI, and they performed a deletion of the LB sequence of the leader region of the FMDO France 2001, and they study the fitness and the growth curve of the virus, of these viruses, mutant or wild type, and they show a similar growth of wild type and leaderless virus in interferon deficient cells like BHK21, but a clear disadvantage of the leaderless mutant in interferon competent cells. After that, a part of the study was performed at ANSES using the in vitro model, so the cells derived from the nasopharynx and grown at the air liquid interface. So, and the cells are DSP for dorsal soft palate cells and RLE for air liquid interface. So in this model, we applied, we infected the cells at a MOI 0.01 with the wild type or the mutant virus or a conditioned medium as the control. And we we monitored and we followed the infection until 35 days post-inoculation, and we sampled every 2-3 days the cultures, and we collected cells and supernatant and so on to be able to analyze this experiment. And we observed that we obtained no persistent infectious leaderless virus by viral isolation. By RT-PCR, we observe only residual leaderless viral RNA compared to the wild type. We observe also that the leaderless viral load was almost undetectable after the early stages of infection, so after 48 hours post-inoculation. And we have first results of the transcriptomic analysis that was performed at FLI. And the first conclusion, and it is still under study, there are many differential expressed host genes between leaderless and wild-type infection at 12 hours post-inoculation. Leaderless FNDV induces interferon beta already at 6 hours, and wild-type does not. And the leaderless FNDV induced IgG peak after 6 hours, and for the wild-type, it is after 12 hours. So it is in line. It was quite expected, but now we have to trim all the data and to refine and to compare. So it is still in progress. And another part of the study was realized in vivo at FLI by in Michael team. And roughly 2 groups of 8 kettles were used. The intranasopharyngeal deposition was used as a way of infection to mimic natural infection. 2 animals per group were euthanized after 24 hours for the early tissue distribution. Clinical exam was done with sedation at different days until the end of the assay at 35 days after inoculation, and many samples were collected from animals. And what about the results? As you can see on this slide, all remaining animals in the wild-type group developed vesicular lesions beginning on 4 days post-inoculation, while leaderless infected cattle did not develop any vesicular lesions or clinical signs of FND. In line, in the wild-type group, a peak of shedding between 6 to 10 days was observed with 2 persistently infected animals, while concerning the leaderless group, no viral RNA was detected in any sample in the course of the experiment. So, to conclude on this part about the relationship between immune evasion and persistent infection, concerning the in vitro DSP model, the leaderless type O virus was shown to be strongly attenuated in this model, cannot establish a prolonged non-persistent infection. And about the role of ALPRO-mediated immune evasion in persistent FNDV infection, it will be completed by ingenuity pathway analysis pending on early time point samples, so 6 hours or 12 hours post-inoculation. And concerning the in vivo part of this work, a pronounced generalized disease and virus shedding was observed in the wild-type group, but no clinical signs of FND nor any detectable virus was observed in serum secretions of tissues for the leaderless group. 2 out of 6 animals in the wild-type group were persistently infected until the end of the assay, and no evidence of persistent infection in the leaderless groups. So overall, this confirms the strong attenuation of the leaderless FNDV for new serotype and the variant panzootic parental lineage, so Panagia. And leaderless viruses could be used for safer production of inactivated vaccine or for virus neutralization test with reduced flow containment requirements. So this study has different conclusion for more basic research, but also for to imagine a new way to perform neutralization test or with reduced beer containment requirements. And if we continue with the work we perform about interaction between FNDV and the type 1 interferon response, and also with a potential link with persistent infection. I will continue and end with this PERSIFA PhD project, so conducted from 2020 until 2023 by Morgan Sari. And he used an interferactomic approach in 4 target species to search for interaction between FNDV O virus, so the same, the O Mayenne 2001, O France 2001, and some protein from the type 1 interferon response for each species of interest. And which proteins targeted? We decided with other colleagues, because it was a transversal translational project in our virology unit. We decided to target 16 type 1 interferon proteins because regarding to the literature, they were shown to be implicated in more than 75% of various host protein-protein interaction. So Morgan used nanoluc-2 hybrid bioluminescence screening to search for this PPI between type 1 interferon pathway and viral protein. So he performed the construction and cloning of the genes encoding these proteins for 4 species. And he performed this nanoluc-2 hybrid bioluminescence screening. So just to be quick, he fused each protein to a part of the nanoluc. So in different combination with the part of nanoluc at the N-terminal or the C-terminal part of the protein of interest. So many construction to investigate, and then the objective was to transfect this construction in cell line, in human cell line 293T, and afterwards to measure the bioluminescent signal and to conclude on this potential interaction. And about the results, Morgan obtained these tables, this heat map, if we can say that. And you have an example of what we shown concerning the interaction between the 3D polymerase of this type of virus and the 16 protein investigated, but Morgan performed this screen for all the 15 protein of the type of virus. But the most interesting results were obtained for the 3D, and the 3D was not described as leading antagonism before in the literature. And he showed different interaction, namely between IKK alpha, IF3, MABs, but concerning MABs was particular of interest because moving to confirmatory test using GST pull-down experiment, he was able to show a specific interaction between MABs from the swine, so from sus profile, and the 3D polymerase, but no interaction when using the version of this protein cloned from cattle, sheep, or goat. And he investigates further the phenotypic impact of this interaction, and perform some Lucifer's reporter essays using the 3D and investigating the induction phase of the type 1 interferon using reporter assay, and he shows that inhibition of this phase in cell line, so in human cell line, but also in cells, primary cells derived from cattle, but also from pig. So the more pertinent cells. So the inhibition was in the range of 40%. And it was in line with with this X map, if we go back, because all these 7 proteins are from the induction phase of the type 1 interferon. So these results were quite expected, quite in line with what we observed with the nanolux screen. So about the take-home message, Morgan showed that a protein interaction between 3D polymerase and the interferon pathway with a particular interesting result with this interaction between MABs and 3D. But the question is still, do these protein-protein interaction exist in infected relevant models? And what are the role of identified PPI in the establishment of various host equilibrium during persistent infection? And in parallel to this work with the screening with 2 hybrid screening, Morgan also developed a model derived from porcine soft palate to be able to compare the DSP from bovine and the DSP from pigs with in mind a new tool to be able to compare what happens after infection and to continue to investigate about interaction between FMDV and host. And so to conclude, and all this work, we can say that protein-protein interaction between FMDV proteins and the type 1 interferon pathway probably. So what role do they play during infection? Do these interactions exist in an infectious context? Is there any variation of this protein-protein interaction according to the strain? So to address this issue, we need ad hoc cellular models and appropriate tools in target species. Other pathways could be involved in the antiviral response and hijacked by FMDV. So several FMDV proteins are shown involved with autophagy proteins, so induction, amplification. And we know that autophagy can regulate interferon response. Autophagy could enable the non-lytic release of the virion from the cells. So with a question regarding the potential role in FMDV persistence. And many FMDV host interactions aim to inhibit the establishment of apoptosis in cells by targeting P53, so another chapter to investigate. The question is also that as the interaction described between FMDV and cellular response pathways sometimes have contradictory effects, it would be interesting to investigate, in addition to the model used, the chronological occurrence of these interactions. Some of them may only take place at the beginning of the infection, while others could participate in the maintenance of the virus host balance in the longer term and potentially in the persistence of FMDV. So overall, studying protein interaction between FMDV and its different hosts combined with transcriptomic analysis in relevant cell models from species of interest could greatly contribute to better understand the pathogenesis of FMDV infection and thus improve FMDV control. And what's next? So just roughly, for next chapter or next seminar in a couple of years, we will continue the PERSIFA project with the new PhD project called FMDV Escape. And this project is performed by Fanny Lambert in my team since the 1st of October about various host interaction in food and mouth disease virus acute or persistent infection. We will continue to improve in vitro model, 2D or 3D cell cultures. And we are to this aim to be able to continue with our model of pertinent cells. We will maintain our collaboration, but we have also initiated new collaboration, for example, with colleagues from South Africa, like Melanie, with Christopher Kasanga, so in Tanzania. And collaboration are more than welcome. So I will finish with acknowledgement because all this work is conducted with a very good relationship with all partners in the world. So thank you to all collaborators. Thank you to my team. And thank you for your attention. And I hope I was not so long. So thank you so much for your attention.
Thank you so much, Sandra, for that fascinating presentation on the host modulation of FMDV during infection. So I'm going to start with a question. In the in vivo leaderless study that you show, do you know if the animals inoculated with the leaderless seroconverted after being inoculated? I am wondering if the lack of persistence is simply due to the virus' poor replication capabilities. We know that leaderless viruses are severely attenuated. So I was just curious if you detected seroconversion.
In this experiment, so thank you for the question. In this experiment, there are typically in this work, no seroconversion was observed.
Yeah, that's what I thought, because we have done a similar study with serotype A, and leaderless is severely attenuated.
Yeah.
My understanding is for persistence, the virus has to replicate at some point. So that's why I wasn't clear whether the lack of persistence detection was basically because the virus is not replicating well in the host.
But you can also imagine that persistent virus will replicate at so low level and probably in structure a bit far from the detection from the immune system. And I remember the works by Juliet and Charleston. They say that the virus could be locked in germinal centers and escape from the immune response. So there could be also a balance between a very small replication in the very small structures and like to be blind to the surveillance by the host.
Thank you. Melanie?
Thank you so much, Sandra. Very interesting presentation. Just a reminder to all the participants, please post your questions in the Q&A box so that we can be able to answer you. So we have a question from Mariano. He says, thank you for a very interesting presentation. How do you relate the transcriptomic, proteomic, or PPI differences found in cell cultures between the leaderless and wild-type FMDV at very early times post-infection, 12 hours, with their ability to establish persistent infections at much later infection times?
Okay, so thank you for the question. So if we go back to the results, just let me check first. Sorry. We saw it was expected that at really early stages of the after infection, it was precisely in this timeline, because after 12 hours you saw almost anything, and as expected, you see the difference between induction of interferon beta by the leaderless compared to the wild type. So it's clearly the induction is clearly delayed. Probably with the leaderless, you have not the capacity to block the interferon response, and probably it is why you allow the establishment of the equilibrium between the virus and the host. For the moment, from these first results, we cannot completely conclude on that. For the moment, the transcriptomics results just are in line with what we expect with the phenotype of the virus, and the deletion of this protein known to have a huge impact on the interferon response. But now we will refine and we'll train the results, because it's really the beginning of the study.
Thank you so much. Giselle?
Thank you. We have a question by Juan Maria Kelly. Thank you for your talk. Have you evaluated if PPI exists between 3D protein as part of the 3CD precursor?
So thank you for this question. So for the moment, no, because it's so the work done, as I told, the work was done on all protein, but as 3A, 3B, 3C, 3D, not all the combination, but it was already a lot, because you have to combine with all the version of the protein from different hosts, and the PhD was only 3 years, so we had to be to go straight. But it's a really interesting question. But sometimes it's also a limitation when you study this interaction with the expression of recombinant proteins, because the 3C is toxic as with the proteolytic activities. So sometimes it's not so easy to manage with that. But in our hands, it could be done, because in these human cells, 3C was not so toxic at we could expect, because in other works we we use the 3C to be expressed as recombinant protein in bacteria, and it was completely impossible to see anything. But in this cell line, the toxic effect was not so huge. So why not? I keep this question because I think, of course, it's important to consider the different steps from the precursor, the protein precursor between until the final protein.
Thank you, Melanie.
We have a question from Muhammad Al-Sisi. He asks, what about delivery of FMD antigen to buccal mucosa with delivery? What are your expectations? And also modified live attenuated?
Sorry, I didn't completely catch the question because they are like an echo. So if you can redo, please, Melanie.
Yes, you can also check the Q&A box, Sandra, if you are not sure. But let me just repeat that. It says, what about delivery of FMD antigen to buccal mucosa with delivery? What are your expectations? And then the second part is, what about the modified live attenuated also?
So I need to read the question because
do you find it?
No, I didn't find it. But what is the question about the live attenuated? What is
I think Sandra, it's referring to the administration route. So what if the leader is inoculated or administered in the mucosal route?
Okay. So thank you for the question and thank you for the clarification. I'm not more molecular biologist than a vet, but I think the route that was used this time was to mimic the natural infection. I think could be tried but probably the could be the same, but I don't know. To be honest, I don't know what I could expect. I think could be probably the same result. So I don't know.
Giselle?
Okay, thank you. So I have a question. During your presentation, you showed the modulation of the type 1 interferon during the infection dynamics of FMDB. Could you also elaborate on the roles of other interferon families in this context, like type 2 or type 3?
Yes, it can be considered also, but with the data we collected so far, and particularly in the in vitro model, we didn't have some hit or some interesting differentially expressed genes, because apart from those involved in the type 1 interferon, the other regulated genes were in connection with the extracellular matrix and also apoptosis. But to my knowledge, and if my memory is good, we didn't have so much result about other family of interferon.
Thank you, Sandra. Melanie?
Okay, Vivian O'Donnell says, thanks a lot for a great presentation. It will be of interest to continue evaluation of the autophagy role on persistent animals. we demonstrated the role of autophagy in an in vitro persistently affected pharynx culture, and we were able to significantly reduce the virus after treatment with interferon gamma. We believe this may be a mechanism playing a role on the non-lytic release of the virus from persistent infected cells or animals. Do you have any other comments that may support the role of autophagy on persistent infection and the non-lytic release of the virus?
So thank you so much for your question and also for your paper that I have on my office since many years, and it's very inspiring. And in fact, it was in my mind because we know that autophagosome have a structure that are almost from the same origin than the structure, vesicular structure used by the virus to replicate. But reading through the literatures, I saw also that for the classical swine fever virus, there is an interaction between MABs and the Blaise-1 to subvert the autophagy for the replication of the virus. And having in mind that we have some interesting interaction between 3D polymerase and the MABs, it is a way that is it matters for us, and we want to explore with this interaction. And I'm pretty sure that something really interesting was to be studied further.
Thank you, Giselle.
Yeah, thank you, Sandra. Since we're talking about non-lytic release, I couldn't help myself but ask the question about exosomes and what role do you think they could play in the persistent state of FMDV in the host or in the cell, in an in vitro cell culture system?
And to be honest, I'm not completely expert with this part of the biology. It's in my mind for this, but in the same plan than autophagosome, I think exosome is definitely a mean for the virus to try to replicate in the cytoplasm without be seen by the sensor of viral piconaviral components. So it's also part of what we want to investigate.
Thank you. Melanie?
Thanks. So moving on to protein-protein interactions, Sandra, what would you say would be the limitations to search for PPI?
So in this, so thank you for the question. In this presentation, I show the results for screening made this nanoluciferase assay, but in the past we also performed a regular, very traditional two hybrid system with the yeast. So in the yeast, we know that it's quite difficult to manage if we consider some membrane protein expressed at the membrane for cell protein. And considering both technique, as you need to fuse your target protein with a part of the nanoluciferase, for example, you have a biais, and you can miss some interaction just because of steric problems. And Also with this approach, you search for like binary interaction, but you can imagine that this interaction will be part of a complex, but protein complex. But you can also imagine that you can have RNA components in your complex. So if you catch something, it was to continue, and if you can complete with other approach like we did with this Lucifer's reporter essay to continue to investigate the type 1 interferon response and to imagine other functional tests to search for the effect of this interaction or to try to block the interaction. But when you find nothing, it's more difficult. But it is the difficulty inherent of every model. And you have also the tag. So the tag has also an impact. But you need the tag because we have not so much tools like antibodies to go further. So the limitation in our case is also the species we investigate, because we know that we have many tools for human, for the mouse, but not so much for other species.
Thank you. Giselle?
Thank you so much. I think we have reached the top of the hour, and I would like to thank you again, Sandra, for giving such a wonderful presentation. I know there's so many factors involved in the modulation of the host during FMD infection, and I appreciate the input that you have provided during this time. Well, thank you also to everyone that participated during the meeting. We're going to be taking a break for the month of January to recharge batteries, and we'll reconvene in February. And with that, again, thank you so much. Melanie, I don't know if you have some closing remarks.
Yes, thanks, Giselle. Again, I echo what Giselle has said. Thank you, Sandra, for taking the time to present to us. I know your schedule has been really hectic, but still you have done this for us, and we really appreciate it. And it was an excellent presentation. And it's our last presentation for the year, so you really closed us off with a bang. And I'd like to thank all the participants who have supported us throughout the year. It was really an excellent GFRA research webinar series, and it was all because of participation. And we thank all our speakers for the year. And we wish everyone a really blessed and wonderful festive season ahead, and see you all next year. And we also want to thank CRDF Global for all their assistance with helping us to arrange the webinar. So thank you to the team there. So over to you, Kayla.
Thank you, Melanie. And once again, thank you, Sandra, for joining us today. This was a wonderful presentation to close out the 2024 webinars. Thanks to all the participants who joined today. You'll see as you exit the webinar, a survey is going to pop up. If you could all just take a minute or two to fill that out, that really helps us to continue to improve on this webinar series, especially as we move forward into 2025. Your feedback is really important and valuable to us. So thank you in advance for filling that out. Thank you for attending, and we'll close out.
Thank you so much.
Thank you. Goodbye.
Goodbye.